Okay, hi everyone. My name is Min Tang, or you can call me Tommy. Now I'm a senior scientist in Shirley Liu's group at Dana-Farber Cancer Institute, working on single-cell transcriptomics and epigenomics. My background is a hybrid of experimental biology training during my PhD at the University of Florida and the computational biology training during my postdoc in MD Anderson Cancer Center. So today I'm going to give you a lecture on single-cell ATAC-seq. By the way, this is my Twitter handle and my blog address. So I mainly tweet and blog about the data analysis and sometimes complaints about bioinformatics. So if you're interested, feel free to follow me or check out my blog. Okay. We know DNA wraps on nucleosomes in eukaryotes, and transcription is tightly controlled by transcription factors. The accessibility of a chromatin region to a specific TF is critical in gene regulation. So ATAC-seq, or assay for transposes accessible chromatin with high throughput sequencing, is a method for determining chromatin accessibility across the genome. It utilizes this hyperactive TN5 transposes to cut the DNA which is open, and it insert sequencing adapters into the chromatin regions. Then high throughput sequencing yield those reads that indicate this region of increased accessibility. As you may know, there are many other methods to assay chromatin accessibility. For example, we have this mRNA-seq or DNA-seq. For the mRNA-seq, it uses this enzyme called microcargonuclease, which preferentially cut the linker DNA. So the end result, what you get is the nucleosome positioning information like this. And the DNase1 is used to preferentially cut the open chromatin. So this is the profile you get after DNase1 sequencing. And for ATAC-seq, it uses TN5, as I just mentioned, and TN5 cut both the open chromatin region and also the linker region. So in the end, you get all the accessible regions in the genome. So why ATAC-seq is, oh, by the way, so because it cuts, the TN5 cuts both the linker region and also open chromatin region. So after you make a bulk, for example, for bulk ATAC-seq, after you make a sequencing library, you can load the library into TapeStation and check the fragment length distribution. So you can do it in the lab, and you will find this pattern. So you should see a peak with smaller than 100 base pair, which indicates the open chromatin region. And then you will see a peak at 200 base pair, which corresponds to roughly the length of one nucleosome. And then 400, which corresponds to the two nucleosome lengths. And after you get the data, you can also do this computationally, because you get that R1 read and R2 read, and then you know the length of that fragment, right? So you can make this histogram computationally, and you will see similar pattern, this periodicity pattern. So there are so many methods for assaying chromatin accessibility. So why ATAC-seq is gaining popularity? So first, it finds all the accessible chromatin regions, i.e. all the transcription factor binding sites in the genome. I have a question. So are they all active sites or repressive sites? Those are open chromatin regions. Any answer for that? So the idea is the repressors can also bind to the chromatin and protect the DNA from being cut by a TN5, right? So those accessible or open regions can be a repressive sites as well. In addition, it's really fast and easy and requires low starting material. In the early days, let's say, for example, ChIP-seq or other sequencing technology requires a lot of starting material. And for tumor samples or samples that are hard to get, it's not possible to do with other technologies. But with ATAC-seq, you can start as low as, like, for example, 2,000 cells. That's why it's gaining popularity. And this is further developed into single-cell resolution, which was commercially available from 10x Genomics. So you can buy these barcoded gel beads from the company, and you can make single-cell suspension or single nucleus suspensions. And what you can do, you can run both of them through this 10x machine. At the right speed, you will see each gel bead will be associated with a single cell within a single oil droplet. And all the PCR reactions will occur within the single droplet. So all the open chromatin regions will be tagged by this same cell barcode for that cell, and which we can separate computationally later after we get the data. Okay, let's look a little bit closer to the library construction. So we know here each bead contains the P5 is the Illumina sequencing adapter, and this 10x cell barcode is 16 base pair, and this is the this sequence, this fragment sequence, it corresponds to the reverse complementary adapters TN5 inserted. So after the first PCR, so the DNA polymerase will extend the DNA from 5' to 3', right? So you extend from here, and then you extend from 5' to 3' in the reverse strand. So this is what you get. And then at the second round of PCR, you use another set of primers. So for example, now you use a P7 is also the Illumina sequencing adapter, and you followed by this sample index 6 base pair. And this is just reverse complementary to the other adapter that was inserted by the TN5. So after the PCR, okay, 5' to 3' fill in the gaps, and 5' to 3' fill in the gaps. And this is the final single-cell ATAC-seq library that you get. Okay, so in last two lectures, last two lectures, you have learned about single-cell RNA sequencing. And let's just compare these two different libraries. So this above is the 10x single-cell RNA-seq library. And here is the single-cell ATAC-seq library. So R1, the forward read for single-cell RNA-seq contains only the 16 base pair cell barcode plus the unique molecular identifier, UMI. It's 10 base pair for version 2, and later for version 3, it becomes 12 base pair. So either the R1 read will be either 26 base pair or 28 base pair. And the R2 read contains the genomic information. And if you compare to the single-cell ATAC-seq library, the R1 read will contain the 16 base pair cell barcode, plus some genomic information there, and also the reverse strand, which also R2 read, which also contains the genomic information. So for single-cell ATAC-seq, it's a real pair end, but for single-cell RNA-seq, actually you only get one single end because the forward read doesn't contain any genomic information. I have a question here, why there's no UMI in single-cell ATAC-seq libraries? So this UMI in single-cell RNA-seq is used to differentiate the PCR duplicates, right? Why we don't need that in UMI in the single-cell ATAC-seq library? Anyone has an answer?
Because we only have 2 copies of each chromosome.
Right, so we only have 2 copies. So anything more than 2 copies, we know it's a duplicate. So we can computationally remove, flag those fragments. Okay. Other methods in addition to the droplet-based are being developed. So similar to like single-cell RNA-seq, there's the plate-based method in which a single cell is sorted into a single well, like for example, into 96 well plate. There's also this split and pool strategy also initially was used for single-cell RNA-seq. So each So you first split the cells into different plates with different barcodes, and you collect them, pull them again, and then split again. So each cell will undergo a distinct path as marked by the barcode added at each step. But the problem is that you need a lot of cells to start with, because each round you can lose some of the cells and it's not commercially available. So it could be like labor intensive. So far, 10x genomics is still the most popular method, because you buy the reagents and the machine, you run through the 10x machine, you're likely to get some good quality data. Of course, it's a little bit more expensive, so but you need to kind of balance. Yeah. Right. So just in the very early days, just for the purpose of developing the technology, people only do single-cell ATAC-seq alone. But the epigenetics should be interpreted in the context of gene expression. People usually are more interested in doing single-cell RNA-seq and the single-cell ATAC-seq in the same tissue or the same sample. So one can split the same sample into two tubes, and one undergo single-cell RNA-seq assay, and the other undergo single-cell ATAC-seq assay. So somehow because they are from the same sample, not exactly the same cell, but still you may be able to map those two data sets together. But early this year, 10x released a commercial solution for single-cell RNA-seq and a single-cell ATAC-seq on the same cell. They call it multi-ohm data, which is also gaining popularity. And we will analyze this multi-ohm data in homework 5. Okay. Yeah. So I will do a first Zoom poll now, and I will stop.